Professor Kell, thank you for joining me again. Today I'd like us to walk through the entire journey, from raw sequencing data all the way to identifying those upstream master regulators. Let's start right at the beginning. Suppose I've just received sequencing data from a ChIP-seq or ATAC-seq experiment. What's my first move?
We begin with read alignment. You take your raw FASTQ files and align them to a reference genome using tools like Bowtie2, BWA, or HiSat2. This step tells you exactly where in the genome each sequencing read belongs. Once the reads are aligned, we move to peak calling. This is where we identify genomic regions, peaks, where your protein of interest was bound or where chromatin is open. Tools like MAX2 can detect these peaks with strong statistical backing. These peaks are your genomic hotspots of biological activity.
So far, that's just telling me where something is happening in the genome. How do I figure out who is making it happen?
That's where motif analysis comes in. We scan the sequences of those peaks for recurring DNA patterns, motifs, that correspond to transcription factor binding sites. This is essentially translating a list of genomic coordinates into a list of potential regulators.
And for that, you need a library of motifs, right? Which is where databases come into play.
Exactly. And here's the big fork in the road. Do you use free resources or licensed professional databases? Let's start with the free route. The public PWM collection from the Gene Transcription Regulation Database is one of the most widely used free motif libraries. It's based on publicly available ChIP-seq data and contains a modest set of position weight matrices, PWMs, that describe DNA binding preferences of transcription factors. You can scan your peaks against public PWMs and get a preliminary list of transcription factors. It's like having a small pocket dictionary. It works, but it won't contain every word you might need.
Once I have this list of transcription factors, how do I figure out what's upstream, what's regulating them?
That's where GeneWays comes in. GeneWays is a literature-mined database that extracts causal relationships from scientific publications. You feed in your transcription factors, and it returns molecules like kinases, receptors, signaling proteins that are known to regulate them. It's a good first step toward building an upstream map. But here's the caveat. Both public PWM collection and GeneWays have limited coverage. You will get only a small portion of DNA binding events and not well-verified text-mined upstream links, so your network will inevitably be incomplete.
All right, now let's switch gears to the professional route. I know TransFac is a big name here. What's the difference?
TransFac is in a completely different league. Instead of a few hundred PWMs, you get over 10,000 high-quality matrices, all manually curated from decades of experiments. But the real advantage comes from something called Composite Module Analysis, or CMA.
I've heard the term before, but can you explain why CMA is such a big deal?
Of course, in real biology, transcription factors rarely work alone. They act in combinations, often binding next to each other in very specific spatial arrangements. This is the syntax of regulatory DNA. CMA detects these combinations, composite modules, in your promoters or enhancers or in the ChIP-seq and ATAC-seq peaks. And this is crucial because a single transcription factor binding site might be common and non-specific, but when several specific binding sites co-occur in the right positions, that's a very strong signal of functional regulation. Think of it like letters versus words. Individual letters, single motifs, can appear anywhere, but specific sequences of letters, composite modules, form meaningful words. CMA finds those words of the regulatory code in the genome.
So with CMA, you're not just matching motifs, you're decoding the grammar of gene regulation.
Exactly. And once you've identified these transcription factor combinations, you can trace them upstream in TransPath, our manually curated signaling network with over 1 million molecular interactions. This step reveals the master regulators, the molecules that ultimately activate those transcription factors. And then comes Genome Enhancer. This platform automates the entire pipeline, from raw omics data to identifying master regulators and suggesting potential drug targets. It integrates TRANSFAC for motif and CMA analysis, TransPath for upstream network reconstruction, and human PSD for linking results to diseases, biomarkers, and therapeutic compounds.
So starting with the public PWM collection and GeneWays gives you a sketch, a partial picture, but moving to TransFac, CMA, and TransPath turns that sketch into a detailed, context-specific map, and Genome Enhancer makes it all seamless.
Precisely. The free tools are excellent for exploration, but when you need completeness, accuracy, and the ability to see causal upstream events, the licensed databases are essential.
Professor Kell, this has been a fascinating journey. From raw sequencing reads to the master regulators at the top of the hierarchy, with CMA as the secret ingredient for uncovering true regulatory logic. Thank you for guiding us through it.
Thank you, Alice. The beauty of this approach is that it lets us go beyond observing effects. We can start understanding causes, and in biology, that's where the real breakthroughs happen.